Dear guests, dear students, welcome to the inaugural meeting of the Minds seminar. We hope to continue with this series. Today, as the first speaker of this seminar, we are proud to welcome our graduate, Furkan Öztürk. We are here in the home of our founder, İhsan Doğramacı and Ayser Doğramacı. We think that this is an excellent opportunity for our students to see the legacy of our founder. So, right at the outset, I would like to remind you that after the talk, please go to the library and see the documents there. We are organizing this seminar to expose our participants to cutting-edge research, the underpinnings of a culture of creativity, and how to build a successful career in research and scientific inquiry. I would like to take a moment and share with you a story now. This is how the meeting of the minds idea came about. Here I would like to draw your attention to Jim Watson and his wife Elizabeth Watson, who were close friends of our founder, İhsan Doğramacı. When Jim Watson and Elizabeth Watson married in the 60s, they came to Ankara for honeymoon. And when we became also close with Jim Watson, he told me that there are three Turks that he admires. One of them is, as you will probably guess, Öztürk. The other one is our founder, İhsan Doğramacı. And the third Turk that he admires is late Erdal İnönü. In 2011, Dr. Watson and Elizabeth Watson spent about 1 week as the guest of Bilkent University in Turkey. And during this week, we became even closer. And actually, the center picture that you see here is Jim Watson, Nazlı Başak from Koç University, and myself in a deep discussion. At that moment, Elizabeth Watson and my wife İclal walked in, and Elizabeth Watson said, "Wow, there is a meeting of the minds. Let's not disturb." And İclal said, "Let's take a picture." And this is how we have the picture. The more important thing that I would like to share with you is what we were talking about. You know, Dr. Watson and Elizabeth Watson married in the 60s, as I told at the beginning. And then they had two sons. One of them is Rufus, and you can watch it. This is public information now that I can share with you. Unfortunately, Rufus is affected with schizophrenia. And during that conversation, we were talking about Rufus. And our discussion was, as we are geneticists, Dr. Watson was telling us that, you know, now, after the discovery of the DNA in the 50s, and we are in the 2000s, now it is well established that such diseases are because of mutations, changes in the DNA molecule. And I would like to bring to your attention three individuals here, Hermann Joseph Müller, John Haldane, and Carrie Stephenson. Müller is a Nobel laureate, and in the late 40s and early 50s, actually he is the one who showed that the DNA molecule indeed changes, mutates. At the time, his contemporary Haldane actually calculated, without any molecular data, just epidemiological observations of phenotypes, what would be the new mutation rate in humans. And Karistafenson, in the early 2000s and 2010, 11, 12, he is the one who directly first observed how many new mutations occur per generation. What I mean is, imagine an individual has a mother and a father, and that individual, when comes into being, has new mutations. The number is 60 to 70. Each individual carries 60 to 70 new mutations that are not present in the parents. And it happens that it happens more frequently in dads than in moms. And Dr. Watson was making the conclusion that the schizophrenia mutation is actually paternal. And he says it is my fault. And actually, since I was so busy working on the DNA molecule, I got married later, relatively. And as you age, of course, these mutation rates increase. I was terribly affected by this. I mean, that night, I couldn't even sleep or walk during my sleep, and started thinking about this over and over and over again. And you know the 5 questions, huh? What, where, when, which, why. Why is the most difficult one to answer. Nevertheless, when you look at a mutation, when did it happen? This is a critical point. It can be before the parents, meaning it happened 500 years ago, 300 years ago, 5,000 years ago, and is carried in the germline of individuals. And I call this pre-parental, before the parents. And there are actually thousands of recessive genetic diseases because of them. Second one, parental. During the formation of the gametes, a mutation may happen. So this is what Watson is thinking about, that in his gametes it happened. So 60 to 70. But there is a third possibility, post-parental, post-zygotic, meaning when an embryo, a zygote, forms single cell, then becomes 2 cells, 4 cells, 8 cells. During this very, very early developmental stages, also a living cell is open to mutations. So this 60 to 70 number, is it really parental, or do we have a proportion that is post-parental, after? So this became a Ph.D. Thesis at Bilkent. And here you see Gülşah Mervedal. And indeed, she calculated this. We used monozygotic twins, twin brothers, and their parents. And what we found out is actually up to 20% of the new mutation rate in humans is actually not parental, but very early post-zygotic. This has, of course, profound implications. Nevertheless, we went to Watson and told him that, there's a 20% chance that it is not your fault either. And it just happened in the parent. And the implications is, all these dominant disorders, thousands of dominant disorders, some of them could very well be due to post-zygotic mutations, and you will never discover them in a routine genome sequencing for health decision making. So, a new area. So this was a meeting of the mind project. And now we are doing another meeting of the mind, and our speaker is Furkan Öztürk. I said at the beginning he's a graduate of Bilkent University. Then he completed his Ph.D. at Harvard University, Department of Physics. In 2021, he realigned or changed his research topic and started working on the origins of life under the supervision of Professor Dimitro Sezlo. And actually, he made a groundbreaking discovery that Nobel laureate Jacques Szostak defines it a real breakthrough in science, which was celebrated in all different kinds of outlets and scientific magazines. He is a recipient of awards, Gertrude and Maurice Goldhaber Prize from Harvard Physics Department, Atatürk Science Award, Innovation Prize, and the JCI 10 Outstanding Young Persons in the World. Right now, he's an independent research fellow at Harvard University and King's College at Cambridge University, working on the same problem of the origins of life. And you all know that he's a very prolific YouTuber and actually has excellent recordings with scientists, his own insights that he shares with the world. On a last note, I would like to emphasize here that all of you, please listen to him with your minds open, because this is a meeting of the mind. We had the most recent meeting of the mind session only 3-4 days ago with Professor Mehmet Sarıkaya and Furkan here again in Ankara. And please become now all ears and listen. So the floor is Furkan's.
Thank you very much. This is an incredible introduction. And I'm so honored to be here and humbled because not just me, but most of my family members were educated here. So we are basically bread and butter Bilkentiers. My uncle, my cousins, and I was educated here. I learned physics, chemistry. In fact, my first chemistry teacher is here, and it's very humbling to speak in front of him. And today I'll share you some of the important properties of genome, and we just heard how important is genetics for life, and I'd like to introduce you my recent research on this topic. And I have somewhat of an identity crisis. I had my PhD in physics, and I worked on a problem on molecular chirality under the conditions of early Earth. So my research spans a lot of disciplines from physics, chemistry to planetary science. And although I am an expert in certain aspects of this problem, I'm by no means expert in all. And without the help of incredible colleagues from all around the world, my research would fall short and I wouldn't be able to complete what I have done. So I really am honored to work with these incredible scientists. So My topic will be on chirality, and chirality is not just a molecular phenomenon, but it's simply a geometric phenomenon. And objects are said to be chiral if their mirror images are not superimposable on each other, just like our hands. And it turns out molecules, if they are beyond certain size, also satisfy this property. This is chirality. And if you look at the manifestation of chirality in biology, in all biological systems, what we see is essential molecules of life. Amino acids, sugars, nucleic acids are all chiral. And they basically can exist in mirror symmetrical pairs as left-handed and right-handed objects. But the interesting thing is that biology is extremely picky when it comes to chirality. It selectively uses left-handed amino acids and right-handed sugars and nucleic acids. And that defines life as being homochiral. So chirality is homogeneously present in biological systems. And the question is why? Like what physical reason, or what is the reason behind this feature of life on Earth? How can we account for this feature at the very beginning of the life, at the origin of life? And Science magazine in 2005 recognized this problem as one of the biggest unsolved mysteries in science, as 125 questions that we don't know. And that's the problem that I'm going to address in this talk. And I will limit my level of detail to very basics because there are a lot of disciplines here. There are experts in physics, chemistry, and genetics, and even philosophers. So I hope that everyone gets something out of it. So I'll hope to not boggle into details that much. So, chirality is a feature of life on Earth, but why do we need homochiral chemistry? Why is it important beyond just being a feature of life on Earth? Okay, so if we just envision a type of life, we have to consider a type of genetic molecule which can transmit information and maybe do some catalysis. And if it can do some useful catalysis, then you need to replicate it as well. So transmitting information and copying useful catalysis molecules requires some type of replication. And molecules like RNA and DNA are chiral molecules. They are genetic molecules, and they're heavily chiral. They are composed of individually chiral units, and they stack on top of each other. And when they want to replicate, they need to base pair with each other. And the base pairing is successful if the strands match with themselves geometrically. Otherwise, due to mismatch, the copying fidelity just goes down drastically. In fact, this has been demonstrated experimentally in 1984 by Jerry Joyce. And what he demonstrated is from a racemic pool of ribonucleotides, you cannot create a functional genome. And statistically, if you just count the number of chiral centers and the number of base pairs, just having a random arrangement of a homochiral genome with high yields and selectivity is nearly impossible. So without homochirality, without figuring out how homochirality arose, we cannot create a functional genetic code, a genome, and without that, we cannot envision life. So it is not just a feature of life on Earth. It is a prerequisite for life on Earth. And so this was kind of a big picture analysis of the problem, and we can further constrain the problem into what stage of origin of life we are considering, right? So in the beginning, there were gases and simple molecules on Earth, like water, carbon dioxide, cyanide, some sulfur, and some energy. And from those, with some chemistry, which I will show in a bit, we can synthesize monomers, most of which are chiral. And after that, we need to have a stage of functional polymers. So these are genome, like peptides and those type of molecules, which are even more heavily chiral because they're now in a helical secondary structure as well as their individual constituents are chiral. And as I just described, their functional properties heavily depend on their chirality. So, and if we just remember our discussion about the chirality of genome, basically we need to figure out the problem somewhere in between the synthesis of monomers before we condense them to make functional polymers. So this is the place where we will try to address our problem. Not afterwards, because before you make them homochiral, you cannot just envision the beginning of life. We can further break the problem up, the homochirality problem. And if we do a standard synthesis of a chiral molecule, in the absence of a physical or a chemical bias, you make the molecules in a statistical mixture of left and right-handed pairs, which is a so-called racemic mixture. And if by some means you can introduce a bias that can couple to chirality of molecules, which can nudge the system into one direction more than the other, you can basically break the chiral symmetry and introduce a little bit of a bias. 1%, 2%, 0.1%, whatever it is. And then this bias can get further amplified, which is now a little bit easier because now you have a direction to go. You broke the symmetry already. You can amplify it, again, by physical or chemical means to realize a homochirality in a compound. But then the problem is, right, like life is not a single compound. It is composed of many different chiral molecules. And either we are going to do this step for each and every chiral molecule or propagate chirality from one molecule to the rest of the network by stereoselective interactions, right? So this step is also necessary. It's often ignored, but it is necessary. If you want to envision a homochiral life at a network level for all molecules, you need to be able to address the chirality of every single compound, not just RNA, not just peptides or like metabolites, okay? So that step is also necessary. Okay, so I will now start with the first part, and in my talk, I'll talk about all aspects of this problem, the symmetry breaking, the amplification, and propagation, and where could this happen on early Earth. So I will provide an environmental basis for our solution, which is also necessary. We cannot ignore Earth. Geology is a dirty discipline. It's hard to work with, but we cannot ignore it. Okay, so I'll start with this symmetry breaking agent. And this is the so-called chiral induced spin selectivity, or CISS, or CISS, whatever you say, effect, which it was discovered not by us. Actually, it was discovered first in 1999, but its discovery became more prominent in 2013 in the group of Ron Nahman in Weizmann Institute in Israel, who was one of my mentors, actually. And the seminal experiment that established this effect is the following experiment. When the researchers take a gold surface, which is not a magnetic surface, not ferromagnetic surface, so it has electrons of equal spin distribution, right? It has electrons are unpolarized, let's say. And they put a chiral monolayer on top of this surface, which is right-handed double-stranded DNA molecules, and then they shine light on the surface. When you shine light on a surface about the work function, you eject electrons. They're photoelectrons. They're unpolarized because they come out of gold. This interesting thing is, as the electrons propagate through this chiral layer, and when you measure their spin polarization after they propagate, after they're transported, they get heavily spin filtered. Okay, so this is the CISS effect, and this happens at room temperature, and the spin polarization is unprecedented levels, near unity spin polarization. In this very experiment, it was 60%. And the interesting thing is when they replace the chirality, molecular handedness of molecules from right-handed DNA to left-handed peptides, the effectively filtered spin state also flips. So what they basically establish is they demonstrate the coupling of chirality to electron spin even at room temperature at very robust levels. So this is the experiment that established this effect. And the manifestation of this is very important, and this is what we utilize. Right, we talked about selecting one chiral molecule but not the other, but this is about selecting spin, not selecting chiralities. How can we connect these? Well, one other manifestation of CISS effect is that when you polarize molecules, when you charge polarize chiral molecules, by, let's say, applying electric field along their molecular axes, you push charge from one side to the other, and you realize a so-called dipole. It's a charge dipole. And this charge dipole is agnostic to chirality. It doesn't recognize if it is left or right-handed. As you see here, you have a left-handed and right-handed molecule. Both have one like positively charged end and a negatively charged end. But when you charge polarize and realize a dipole in a chiral molecule, what you do is you transport charge in a chiral medium. You transport charge from one end of the molecule to the other, which is spin dependent. Okay? So any charge polarized chiral molecule also gets spin polarized. And that is the key physics aspect that we will use. So essentially, non-magnetic chiral molecules can become transiently magnetic if you polarize them. Right? It's a transient effect. And we utilize this. And the way we utilize this, well, if they get polarized, you can select them. If the molecules become magnetic, you can filter them with magnetic interactions. Where can the magnetic interactions can come from? Well, they're magnetic surfaces. Magnetic surfaces such as iron oxides, which were very abundant on early Earth. In fact, it's the most abundant natural magnetic mineral on Earth is magnetite, iron oxide. And what we did is we basically studied a surface process on a magnetic surface, a magnetic iron oxide surface that involves an equal mixture of left and right-handed molecules. They're quite important molecules, but I'll describe their importance for prebiotic chemistry or for origin of life later. Let's first just look at the physics of what we have seen, then we will talk about chemistry. Physics is always first. So when we started with a mixture of left and right-handed molecules, as these molecules approach the surface, they basically undergo charge reorganization due to dispersive forces that between the molecule and the surface. And what you do is, again, you transiently charge polarize these molecules, which gets accompanied by spin polarization due to the effect that I just described. And then one molecule becomes up spin on one end, and then the other becomes down spin on the other, right? So now you basically break the symmetry, the spin symmetry in these two different molecules. And because the surface is magnetized, it pulls one enantiomer, one-handedness, and pushes the other one. And you effectively increase the concentration of one more than the other. And the crystallization of one enantiomer becomes faster. It's a kinetic phenomenon. It's not a thermodynamic phenomenon. So when you wait ad infinitum, you get a 50-50 mixture, but you can do this in a flow setting. And if you keep the solution always racemic, you can crystallize always one enantiomer. And on the right, what you see is RAO molecules. It's a precursor of RNA. It stands for ribosaminoxazoline. It's a ribose-like, nucleotide-like molecule on a magnetite surface. And when you pick these crystals and dissolve them and analyze their chiral properties, what you see is they're heavily enantioenriched. And here I show you a circular dichroism spectra of the crystals that form on a magnetized surface in solution at room temperature. Nothing fancy, no vacuum, nothing. And what we see is in a control experiment where the surface is non-magnetized, we don't see any chiral selectivity, which is the absence of circular dichroism signal. And when we magnetize the surface in one direction, we get one-handedness of the molecules. And this is like not just one molecule, but a mixture of molecules, right? And when we flip the magnetization, the more effectively crystallized molecules becomes now the opposite-handed. So you control which enantiomer crystallizes faster by just switching the magnetization. And recognizing that early Earth surfaces will not be 100% spin polarized, we also studied the opposite of this effect. Right? Typically, you will get a statistical mixture of spins on a surface, and you need to purify the surface to get a better and better effect. But due to the same effect, what happens is if you crystallize a homochiral molecule now of one-handedness on a molecule with no net magnetization, when you crystallize this molecule on the surface, underneath the molecule, you see magnetization. which again depends on chirality. When you flip the handedness of the molecule, the magnetization direction flips, right? And we measured this using polarized light in a so-called Kerr effect microscope, which you see on the right. And very recently, like a few weeks ago, we submitted 2 papers that studies the temperature dependence of this effect with Yael Capon from Jerusalem. And here, it's actually beautiful physics, which I won't go into detail, but what we see is we saw a quantum effect, which is the CISS effect, that gets stronger with temperature, which is not unheard of, but it's rare, right? Which also shows you that the induced magnetization by chiral molecules will be robust across a range of temperatures that would likely be present on early Earth. It's not a zero temperature effect. It's actually stronger at higher temperatures. Okay, and of course, like it is no-brainer that you can envision these 2 effects, meaning induced magnetization by chiral molecules and selective crystallization on a magnetized surface together in a so-called feedback, right, in which the first crystallized molecular layer magnetizes the surface underneath due to effect number 1, and then due to effect number 2, you get faster crystallization of 1 enantiomer nearby these areas, which then amplifies the effect because it will distribute the spin to these sides, right? So it is a self-reinforcing feedback which spontaneously breaks symmetry, and the direction that it will go can be tipped by a small magnetic field. Okay, and this was just the mechanistic aspects of this study, which you can compare actually the previous attempts to break chiral symmetry. And so far, I talked about amplification as well because of some special crystallization properties of our molecule. But if we just think about chiral symmetry breaking, the study that we have done supersedes the previous attempts due to prebiotic plausibility, due to the robustness of the effect at room temperature and being in solution. And you can check the references, but there are previous attempts, of course, such as using the parity violation in fundamental physics, which seems to be orders and orders of magnitude weaker than room temperature, which is hard to realize in real systems. And there is, of course, SOI reaction-like systems that people study, but so far there are no prebiotic analogs. And Wiedemann ripening seems to be an interesting attempt as well, but it is not a deterministic effect, and it requires solution phase racemization and careful control of crystallization parameters. And circularly polarized light is absent on early Earth. It's only in solar nebulae. Okay, so now I talked about actually most of the experiments and most of what I've done, apart for some aspects, but I decided to do this talk in this type of way, meaning if you just talk about the mechanism and show that it works, is that enough, right? Okay, like if you look at just the numbers, that it's much better, much robust, etc. mechanism, is that enough, right? I argue that it's not, and we need to consider more aspects of the origin of life's chemistry and the plausibility of the geochemical scenario. So now I'll go to those parts. So I talked about the mechanism. I said, right, it's the chiral-induced spin selectivity on magnetized surfaces, and crystallization solves the problem of chiral symmetry breaking. But in what chemistry, with what compound, does that matter? How do you propagate the attained chirality in this molecule, which seems to be a random choice to the rest of the molecules? And how can we envision the system on early Earth, right? So now I'll talk about those aspects. So what kind of prebiotic chemistry should we consider? Okay, and how am I doing on time?
You have 20 more.
20 more minutes, perfect. Okay, so you may have heard of Miller-Urey experiment, right? Miller-Urey experiment, 1953. You take a gas chamber of reduced gases, like ammonia, methane, and you do a spark discharge experiment. You excite all the molecules to their excited state, And when you like do that type of high energy processing, everything gets out of it. You know, you kind of occupy the phase space of all molecules at their ground level. It's not a selective chemistry. It's not a high yielding chemistry. It typically creates a mess, and the mess contains a little bit of useful stuff, such as glycine and alanine, but not really a good way of doing chemistry, because then you cannot further utilize them. A better way of doing it is doing selectively and with high yields. Because if you don't do high yields, and if you have a multi-step synthesis, typically your yields converge to zero. And this type of network is, in my opinion, a far better way of doing chemistry, which is so-called the cyanosulfidic chemistry, which is discovered by the group of John Sutherland at Cambridge University, who is my close collaborator. And in his network, what he does is he starts with hydrogen cyanide. It contains a triple bond. It's a high energy molecule. And it does nitrile chemistry with some subtleties using UV light, which would come from solar light, and creates all nucleobases, nucleotides actually, and 8 amino acids and precursors of membranes, phospholipids, in a shared chemistry with subtle transformations under the conditions of early Earth. And this type of network, of course, I won't go into details, is both selective and high yielding. And very interestingly, it makes 8 amino acids. And what you see here is sparks the eyes of Typhon here, because it's the genome, it's the genetic code, right? It is the alphabet of life. And it has amino acids that correspond to certain anti-codon sequences. When we look at these, there is some oddities in these alphabets. Some amino acids are more abundantly represented. They're the family box amino acids that are 4 anti-codon sequences correspond to them, and there are 8 of them. Interestingly, this chemistry produces 8 amino acids, and without doing this by design, these amino acids happen to be the ones that correspond to family boxes. It's quite a coincidence, right? Like you make the ones that are most abundantly represented by the genetic code, which happens to just come out of chemistry. It's a good indication. It's not a final proof, but it's a good indication that it's on the right track. And the molecule that we work with is a central molecule of this chemistry. I said that it starts from hydrogen cyanide by some reductive homologation chemistry. The details don't matter. Chemists will deal with them. You make glyceraldehyde first, which is the first chiral sugar, an important molecule. And then by incubating this molecule with another compound that you can make from glycolaldehyde and cyanamide, you make pentosaminoxazolines in a racemic mixture. And then one out of those 8 molecules of also enantiomers is ribose aminoxazoline. It is the ribose-like sugar, so it is the right steric form for making ribonucleotides. And it is a direct precursor of RNA and DNA nucleotides. When you make this molecule in its homochiral form, you can make rest of the nucleotides in their homochiral form. This molecule crystallizes as a conglomerate, and that's an essential physical feature for seeing what we have seen. It is very insoluble, meaning you don't need high concentrations of this molecule to crystallize it. And it doesn't racemize in solution, unlike glyceraldehyde, which undergoes aldol chemistry. And it also doesn't isomerize. So once you lock the chirality of this molecule, you basically figure out the chirality of genome. It's kind of at the bottleneck. And it satisfies the right physical properties and chemical properties that matches with our mechanism, right? This is this is an organic synthesis thing, which I have no idea about. And what we discovered is a physical mechanism which seems to be a perfect match with this molecule. So that's why we work with this molecule. And when we attain this molecule in its homochiral form, we can safely say that we can make RNA and the genome in its homochiral form. That's why it is very appealing. Okay, well, you may have been thinking, well, this guy said amino acids, peptides, metabolites, and now he's talking only about RNA, right? So what about that? So how can we propagate chirality of RNA, the genome, to the other molecules, right? To the other classes of compounds. So let's deal with those. So there's an interesting discovery in the last few decades by several groups involving Jack Szostak that Tayfun Hoca mentioned in the beginning, also John Sutherland's group, in which they study formation of coded peptides on RNA templates. So this is coded peptide synthesis. You first aminoacylate an RNA, and then you look at some loop closing ligations or some other like transfer chemistry in these systems. And what people have observed is if you have a racemic mixture of amino acids, an equal mixture of left and right amino acids, and if you make peptides on RNA templates, the chirality of RNA dictates the chirality of peptides, which happens to match in the direction that life has, meaning right-handed RNA sequences make left-handed peptides almost 10 times faster, more effectively than right-handed ones. And you can actually flip this. It is this paper right here. If you use left-handed genome, you can make right-handed peptides. So you can use RNA to propagate chirality to amino acids and peptides. And it's a general solution. You don't need to like consider, oh, you need to do this for alanine and you need to do this for serine. No, it works for all amino acids. It's quite effective. The stereoselectivity is more than 10-fold. It's almost reaching to 200-fold. It's very high-yielding chemistry.
Is it structure? Is it purely structure?
Well, the chirality is controlled by the chirality of the sugar of RNA. And this allows for propagating chirality to peptides, okay? So nucleic acid seems to be a good place to establish chirality, which doesn't work for metabolites, for instance. Let's look at that. Okay, so here what you see is Krebs cycle, right? It's the kind of an engine of life. It's abundant. And in metabolic cycles, there are two chiral compounds, malate and isocitrate. They're both chiral. One is left-handed and the other is right-handed. And you can actually reduce tartrate to make malate, which enters Krebs cycle. And tartrate also is an interesting molecule, which Pasteur worked with to discover chirality. And it also satisfies the same physical property as RAO. It's crystallized as a conglomerate. In fact, that helped Pasteur to discover chirality. But so essentially this molecule may work for our mechanism, okay, but then when you reduce it to make malate, once it enters the Krebs cycle, let's look at what happens to it. It either dehydrates to fumarate or oxidizes to oxaloacetate, both of which are achiral. Okay, so it loses its chirality. You can make this molecule homochiral, but you cannot propagate it. The same fate is shared by isocitrate. It loses chirality to alpha-ketoglutarate or citrate, right? So without enzymes that control these processes, non-enzymatic Krebs cycle is a chirality grinder. You can inject chirality in it, you cannot propagate out of it. It's not a good place to establish chirality. What about amino acids? Can we go and ask individual amino acids if they want to be homochiral? Well, some of them satisfy the physical properties that we desire, but they all have idiosyncratic chemistries, and you need to go and figure out their chemistries and how they become homochiral one by one, which is not a very elegant solution. It's tedious. And then you need to figure out how can you propagate that to genome and to metabolites. So doing the way that we did, starting from genome and then going to metabolites, seems to be a more elegant way, right? So it's by doing coded peptide synthesis on RNA sequences that you can propagate chirality from RNA to peptides. And then peptides make enzymes, and when you control metabolic cycle with enzymes, they're all stereoselective now. Now this is not non-enzymatic, but the stereoselectivity is controlled by enzyme catalysis. Right? So this way of doing it, meaning this direction, is much more effective than doing it the other way around. Because metabolism by itself is a chirality grinder and you cannot propagate it backwards. And this scheme would be very elegant, right, if we could make the genome homochiral, because then you can just go all the way in. How can you make that homochiral? Well, if you make the general precursor of genome homochiral, which is RAO, then that would make all genome homochiral. And that's what we did. That's what we did on magnetite. So we started from magnetite, which is a physical, with a physical effect, we transferred the chiral information from mineral, from geology to organic chemistry. And by doing some organic synthesis, we transferred that using John Sutherland's chemistry to RNA, and then through coded peptide synthesis to peptides, and then to enzymes, and then with stereoselective enzyme catalysis to metabolites. So now you can see why it's RNA and why it's genome that is important, and why we worked with this molecule. Because once you make this molecule homochiral, you can propagate it all the way around. And this is interesting because you may recall the central dogma of molecular biology, right? It's the transfer of sequential information, which tells us that you can propagate information from genome to proteins, but not backwards. It's the same way around, right? You cannot propagate it backwards. So it is almost central dogma-like information flow, but it's for chiral information, not for sequential information. So it's the right chemistry and right compound, I argue. And now the last bit is the dirty bit, which is the environmental part. I assume I have like about 10 minutes? Okay, perfect. So how can we envision this physical chemistry of chiral symmetry breaking and the organic synthesis under the conditions of early Earth? Well, there are these type of lakes. They're shallow, closed basin lakes with minimal outflow but inflow. That includes a lot of iron oxide minerals. And there's an expert of iron oxides here, which is very good for us. And these type of lakes are discovered in Mars. In fact, Gale Crater is one such lake. It is a so-called redox stratified lake. There is a layer below which there's reduced iron species, which is magnetite, above which there's hematite, which is the non-magnetic iron oxide. And these lakes contain these magnetic inclusions in silica matrices, right? And they're very shallow lakes. When I asked how shallow it is to John Grotzinger, who was the head of the Mars mission at Caltech, what he told me is like, well, you can walk in Gale Crater without your knees getting wet. So it is really shallow. And you can have an inflow in these lakes, and in these type of environments, there's typically a planetary magnetic field which magnetizes these sediments statistically toward one direction. And if you have a flow of racemic material on these surfaces, you typically tend to crystallize one enantiomer faster than the other. So the environment is the shallow lakes with magnetic sediments, because you need to be able to have magnetic minerals, and you need to be able to concentrate materials by wet-dry cycling, because you need to increase concentration for crystallization. And also you need some type of flow to keep the system out of racemic equilibrium. And we actually studied the synthesis of magnetite in these type of environments, which I won't go into detail. This is still work in progress that I'm doing right now in collaboration with geochemists. But in the initial experiments, I used synthetic surfaces that I have fabricated through methods that don't involve prebiotic chemistry. They were basically physical chemistry methods. But now we realize, or not realize, but we basically acknowledge the fact that we need to transition to more and more real materials. So now we make our own rocks almost. And we will study those processes on more and more real surfaces. And the way we make these minerals is we start from an iron carbonate solution, which typically represents early Earths like oceans or like lakes. It, of course, involves other materials such as silica and others, and we will involve that complexity. And when you irradiate this type of solution of iron, dissolved iron, with solar irradiation, you can do xenon lamp, which is almost like early sun, and oxidize iron to magnetite. You produce single domain magnetite, which can get magnetized quite strongly under early Earth's magnetic field because it has very low coercive field. This type of field, it's called coercive field, characterizes how much field you need to demagnetize or magnetize this type of mineral. So it's important metric for us to understand how likely we can get high spin polarization with these minerals. So we make those under the conditions of early Earth. And when we compare their magnetic strength to those that we studied with chiral molecules on top, the one you see here was from our previous experiment, in which what we have observed is the increased coercive field by the introduction of chiral molecules. We saw an increase of coercive field by 1 millitesla by chiral molecules. And when we look at the distribution of magnetic particles that we make in the lab with prebiotic chemistry, excuse me for that plot, it looks complicated, but what that tells you is that there's a distribution of particles that are concentrated here whose coercivity is below a few millitesla. What that means is if you want to flip the magnetic field of these minerals, the magnetization of these minerals with chiral molecules, we can. That's what it means. And then what we did is, this was one project, and then the other project was to understand if natural lakes get magnetized at all or not, right? How much magnetization can we get under real conditions? For those, we went to like lakes. And when I say we, I mean Roger, because he's a geologist and he does more dirty stuff than I do. And we went there, we collected some sediments from this pond in Cambridge, Massachusetts. And when we settled those sediments in a beaker, which is under Earth's field, it involves all the dirty stuff of, like minerals and maybe some biology even. And when we kind of immobilize the sediment in some epoxy matrix and almost like cemented this type of sediment and looked at its magnetic inclusions with a technique called NV magnetometry, which can measure very sensitive magnetic fields. The details are not that important. What we discovered is, of course, that this sediment involves a lot of magnetic particles and inclusions, which are these like black spots in the sediment. And the interesting thing is you can look at the orientation of these magnetic inclusions. And these are like just individual dipoles in the sediment. You can basically project that 2D map into a 3D one, which is a stereogram that represents Earth. You basically see a clear bias toward the direction of early Earth's field. And when you do statistical averaging of these individual dipoles, just as we envisioned, there is a bias toward the magnetic field direction. It's a trivial result, right? It should have the bias.
I can't stop. I will ask a question. Are these bacterial origins?
Okay, good. Some of them are magnetotactic bacteria, dead bodies. Yeah. And some of them are not.
Because that's not unusual.
Yeah, it's not. Yeah. Great question. Yeah. And with this, I basically summarize my talk, right? We worked on a physical mechanism of chiral symmetry breaking on a molecule that is central to prebiotic chemistry, which allows for propagation of chiral information from a single compound to the genome, then to peptides, then to metabolites, under conditions of early Earth, in early Earth shallow lakes with iron oxide inclusions. And that is my summary. And I argue again, whenever you see this type of study, you need to think of all the disciplines coming together, the mechanism, the chemistry, the geology. And of course, I couldn't go into details, but there are a lot of papers that came out of this project, and I kind of summarize them here if you want to look at them. And some of them are very fresh. They're in archives, so you can look at them there. And with that, I conclude my talk. Thank you for having me here.
Firstly, I'm Aparakis. I'm from Math Department. My question is, if the Earth's magnetic field, if the early Earth's magnetic field was reversed, then could we expect the chirality of the organic molecules to also be reversed?
So that's a good question. Actually, I I kind of thought about mentioning it. I forgot, so thank you for asking that question. So it's true that early Earth's magnetic field reverses on planetary timescales, okay? So that's something we need to appreciate, first of all, which is about like 150,000 years, which is not too short, not too long, if you kind of think of billions of years of Earth. But we deal with chemistry here, right? We deal with prebiotic chemistry, typically like nitrile chemistry. Hydrolysis of cyanide at room temperature in water is about 100 years. So the time scale that we deal with chemistry is much shorter than the flipping time scale. So we typically have a uniform magnetic field. And then the other thing is, it's something that I didn't go actually into. When the material magnetizes under Earth's field, it is harder, much harder to demagnetize it when you flip Earth's magnetic field because of coercive field. And when you take a material, magnetize it, in order to magnetize it in the other direction, you need to apply 20 times, 100 times stronger fields. So typically, like, these materials remain robust on the course of Earth's history. In fact, there's a field called paleomagnetism, which uses the magnetic inclusions to track Earth's magnetic field flipping history. If it would flip, right, what we would see is the most recent magnetic field direction. Yeah, but it's an excellent question, and I think that because of chemical reasons and then it's material science reasons, we don't think it will be a problem.
Thank you so much for a great presentation. I'm Ismail from Chemistry Department. Actually, I was wondering the kinetic limits of this enantiomer enrichment due to like CISS effect, because like as far as I remember, it was like 60% around enantiomer enriched, the oxazolone, and from synthetic perspective, yeah, it's enantiomer enriched. But I was wondering, like, this 20% of other enantiomer, is there another mechanism to eliminate that, or like is there a kinetic limit specifically?
Right. So the kinetic part is we haven't studied the time scale in detail, but what we have done is like we looked at certain time points. And after a while, like if you wait everything to crystallize, which is typically far longer than the material that we work with, we typically work with around 10 to 15% yields to get the highest selectivity. And when you reach about like 6 to 70% yields, your enantiomer selectivity goes down to like 10%-ish, which is the like time that I stopped. And there are like some chemists that actually are thinking if you can couple this to racemization of the material that makes RAO to basically realize something like dynamic kinetic resolution. But the way I think of it is to do this in a flow setting in which you always have a racemic material in the solution, and the material that you produce is in the solid phase, and you wash away and dilute the supernatants, and you use the crystals to do chemistry, not the solution. Again, I didn't talk about it in detail, but we actually went to full homochiral, not 60%, because what happens is because of this feedback effect, if you do crystallization again with the same material that crystallized first, you actually go to 100% directly. Like you start from 0, you first go to like anything between 40 to 60%. If you repeat this experiment many times, there are a lot of statistics that we have done. And then if you repeat this again, you go to 100% EE. So that's why in the paper we said in 2 crystallization steps, we went from 0 EE to 100% EE. Yeah, thank you. But if you have any ideas about dynamic kinetic resolution, please share us. I'm scared now.
No, easy question. So, Emre Hözensoy from Chemistry Department, thank you for the nice presentation, Furkan. So, in your talk, in passing, you mentioned that, while you were talking about the adsorption of these different types of enantiomers on magnetite, you said it's not a thermodynamic effect, but rather a kinetic one.
Yeah.
First, can you just can talk about that? What do you mean by that exactly? Another thing is like, in terms of a simple adsorption process, if you just look at the adsorption energies of those 2 different types of enantiomers on spin polarized surface, like a ferromagnetic
Yeah.
material, like a magnetite, whatever. So in terms of order of magnitude of interaction, kcals, kjoules, whatever. So what is the difference between, for instance, those two different types of enantiomers and their surface interaction energies? So you didn't mention any kind of numbers there.
I didn't, yeah. It's actually has been...
That converts into surface residence times, right? So
That's right.
Well, so, okay, I got that the mechanism, the phenomenon is there. But whether its magnitude is enough to explain all of these,
That's right.
selectivity and chirality, you need to talk about the order of magnitudes to say that whether this mechanism could be responsible for the biological reality that we see, right?
That's right. So after our paper, there has been a study in Jülich in Germany, in which they did a very careful study with a lot of statistics to measure the energy difference with cobalt surfaces and other type of chiral molecules. They measured it to be about 10 millieV difference between like right and left molecules. I estimated,
It's less than RT, basically.
well, on order, right? Like it is still, you can see it. I estimate it to be like around 50 millieV. And
Because, the Boltzmann distribution and everything, you're talking about RT,
That's right.
That's
On
enough
order
to
of
beat
room
the
temperature.
entropy?
Huh?
Would that be enough to beat the entropy?
Well, it is, if you look at the Boltzmann distribution, it's like e to like 10 to 15 over 26 millieV. There's still like enough distribution to see it. And it's also, as I said again, in the first part of the question is, what I meant was there's no absolute energy difference if you wait ad infinitum. The reason is the selectivity is from the transient spin polarization as the molecule is nearby the surface. What happens is, if the solution is not racemic, if you start from 50-50 mixture, right? And if you start crystallizing molecules, you start crystallizing one enantiomer first, meaning faster. And then after a while, what happens is you deplete the other, you enrich the opposite enantiomer. So when the concentration difference beats the energy difference, you basically start crystallizing the other one, which starts decreasing your EE. The solution to this is, do this in flow. If the solution is always racemic, you can crystallize ad infinitum. So that problem is related to that. So regarding your first question, so that was for heptahelicines on cobalt. There was another study in Jerusalem in which they did AFM measurements with like surface being magnetized. They come with an AFM tip that is functionalized with a chiral molecule, and they come with this way, and then the other magnetization, and they measure the force constant for one molecule, for one magnetization and the other. In that study, they use polypeptides, and they measured the energy difference to be 100 millieV. So that was another study with AFM. The first study was done in vacuum with, again, cobalt and heptahelicines. So the energy difference, we think...
AFM work is in water, right? It was in situ, probably.
It's in situ, yes, it's not in water. So the energy difference should be on the ballpark with like room temperature between 10 to 100 millieV. And it depends on the molecule and the surface.
Also, Ahmed from the chemistry department. I have just a quick, simple question. So, this process that here you showed us, it happens, on early Earth. But would this leave enough time later for complex biological life to form?
Okay.
The time scale enough? Would be enough after the chiral molecules?
Well, yeah. So. Okay, well, I mean, what we know is it happened. So what we try to study here is like in the stage of prebiotic chemistry, before we even talk about protocells and like last universal common ancestor. In the chemistry stage of like short time scales of maybe 10,000 years, how can this process like give rise to homochiral molecules? And of course, like this is probably like around 4.2 billion years ago, maybe 4.3, maybe 4.1. Right before we saw the earliest signs of life, this should have happened. And we kind of estimate the first like cells to emerge in about like 100 million years after. And then the proliferation of life and the complexification takes much, much longer time, right? Like, it almost seems like once the conditions were ready on early Earth for chemistry to make these type of complex molecules, it happens almost right away. It almost happens right away because after a few 100 million years, we see complex like fossils of stromatolites in Western Australia. It is kind of strange to me that, you know, once chemistry is ready, it makes these molecules in about like 4 billion years ago, but it takes almost 3.5 billion years for complex life to start, right? You need to go to Cambrian for 500 million years. So like, when I gave this talk to like geobiologists, one of them told me that like, origin of life is an easy problem. Origin of life, complex life is a harder problem. So I don't know. Yeah, the complexification of life is hard. Yeah. But it happened, so I guess there's enough time. Yeah.
Well, thank you very much for an excellent presentation. My name is Tuva. I'm not a student anymore. I wish I was. I'm a graduate of electrical engineering. I wonder, have you considered in your experimental setup the effect of gravity on the enantioselective crystallization?
Oh, right. I know why you asked that.
Yeah?
Because you come from...
In microgravity,
right,
you know, the crystals grow better and bigger.
No, we have done all these things under ambient conditions. All of them at like regular gravity, regular field strengths. Maybe you can do it.
Oh,
when you go again?
I would be delighted, yeah. Before I go, I mean, we would be very interested in carrying out the...
But I mean, okay, gravity matters.
Yes.
Because you need to concentrate materials on your bottom of your legs, right? And like when crystals nucleate, they need to fall down to concentrate materials on your like kind of lake sediments. So I think gravity matters, but yeah.
Does that have the presumption that the chirality took place on the planet, not some extraterrestrial comet and came with that?
Yeah, I always like think about terrestrial origins. I think like when you can't think of it the other way. I don't think there is enough evidence to consider that. Also, like you push the problem in a place you know much less about. Of course, you can't rule that out. But, yeah.
And also, just one follow-up. In your paper, you talk about some twinning effect in the crystals, right?
Yeah.
And on those tips, the percentage that you would get would be less, I think, the less
wow,
chirality.
You have read it so carefully that I'm impressed, yeah.
And in microgravity conditions, you would not get those rosette-shaped crystals, but more diamond shapes. Maybe it could help your analysis as
well. Well, okay, that's an interesting, very detailed question. Yeah, the twinning effect was a problem. We solved it in the paper with chemistry. Yep. We actually solved it like very recently by involving a little bit of different chemistry, by involving sulfur, like sulfides, which happens to like bind with the, so sulfides, you need them, okay, for chemistry. We didn't have sulfides in our solution because we didn't see an obvious effect. When you have bisulfide in the solution, bisulfide, these materials, these organic compounds form adides. Bisulfide in our solution, we just solved the twinning problem in a more elegant way than we did in our previous paper.
But I think at the same time, it's an excellent opportunity for us to discuss further more. So I think there should be no time limit on that.
So I have 12 questions.
12. Good.
No, I'm just kidding. So first of all, I want to clarify that one of the first organisms that produce organelles is magnetotactic bacteria.
That's right.
And that actually evolved 3.8 billion years ago. And if you go out and find a small pond, you will actually find 15 different magnetotactic bacteria species.
Yeah.
And on the northern hemisphere, these magnetotactic bacteria see south
Right.
because they want to live under anaerobic conditions.
Right.
On the south, they actually see north,
Right.
the same thing. So this, you don't have to change the Earth's magnetic field to look at these particles.
That's a very interesting observation, right?
That's right.
Like that.
And the other one, I think that will help you write your papers, is that all of those magnetic particles have biological origin. None is non-biological.
I wonder.
I'm just going to claim it.
All right.
If you are right, I'll take you to dinner. If I'm right, I'll take everybody.
No, I think the minerals that whatever the origin of magnetite in our sediments, we don't really care because we make them abiotically anyways in the lab, and you compare those to. I have a question.
Yeah.
When the bacteria makes magnetite, do you see a difference in morphology, or do you just see?
Of course.
Okay. Well, that would tell.
It just turns out that when I met the student who first discovered the magnetotactic bacteria, it was Frankel's student, Brzezinski. So that was the first bacteria.
Okay.
But there are now, I think the last I heard, there were 50 different species.
Okay.
And there are probably 150.
Yeah.
Every single species makes magnetic particles of different morphology.
I see.
They're all magnetic,
All
uniform.
Right.
Now this comes from another important issue. I love your talk. We either have to work together,
Or becoming
like brothers, or I'm not going to see you again. The reason is because those particles that are formed, you're showing it here, right?
Right.
So those particles, you didn't show the crystal structure. I would love to see the crystal structure. You can answer all of those questions.
Oh, I can show the XRD.
Yeah, some other time. So that's coming from the fact that the peptides are interacting with the mineral in a different way, not just during the nucleation, but during the chlorophyll stages.
That's right.
That's where the morphology is coming from. But that's not the question I was going to ask.
Okay.
So if you go to the last slide.
Okay.
Yeah, I love that slide.
The summary slide.
So you're not saying, I discovered the origin of life.
No.
What you're saying is that I discovered a pattern that would lead us to the origin of life.
Of course, this is a constraint.
Exactly.
Right? This is not the origin of life.
And this is beautiful. It reduces to just one single mineral.
Yeah.
One single mineral. If that's true, we are seeing the Nobel Prize winner in front of us now. I'm not kidding. I have seen the last Nobel Prize winner, David Baker, is a friend of mine at University of Washington. So I can see what's coming up. Now, my question is, when I look at this progress,
That's right.
I see, well, someone else should have thought about this.
Right.
It's so obvious. I mean, it becomes so incredibly
Right.
beautiful and
know.
obvious
Right.
now.
Right.
Because if you CO4, sure, it's going to produce a chiral molecule. I don't know those stages, but it goes to RNA.
Right.
Then it produces the L-peptide, L-enzymes. After that, it's a material science and materials chemistry.
Right.
Of course, it's going to produce D whatever, right? D sugars, D carbohydrates, everything else. And this has to be extremely specific. You can't just have left hand and right hand, right? You can only have right hand, and then all of the molecules that are being produced by these enzymes have to be left-handed, but with a very specific chirality. I mean, you already know this. I'm just repeating what you know, what I understand what you know.
I can maybe elaborate a little bit on like why people have.
Then I have my main question.
Right, my people. Okay. why people haven't like thought of it. That's my guess. So it relates to Emrah Hoca's question, the energy differences. It doesn't make sense. People try to crystallize things and get something homochiral out of it. Vietnam ripening was one example, but it's like stochastic, so it produces left and right. Whenever you repeat the experiment, it doesn't produce a deterministic bias. And then people, of course, like thought about magnetic fields, which one of them was Pasteur. The other person was Pierre Curie, right? He tried with magnetic and electric fields. What they didn't think about is the importance of surfaces, first of all. It's not a magnetic field effect. Magnetic field is not a chiral entity, right? It needs to be a surface. And then for the energies to make sense, we need to go to CISS effect. So CISS effect was discovered in 2013. Okay? So 2013 is not that far ago, and it hasn't been recognized even now. Like people don't even know, like maybe you're in surface science community. How recognizes CISS effect as a mainstream field? It's not that much, right? It's a niche community. People haven't like really connected CISS effect to like organic chemistry. And organic chemistry that we talk about, cyanosulfidic chemistry, is 2018, 2016. So like it takes like 5 years, 10 years for like discoveries to like mature enough that people can connect things. And then, you know, the spark in our heads was like when we saw these like lakes with magnetic sediments. I wasn't aware of them. I come from physics. I wasn't aware of this cyanosulfidic chemistry before I went to Harvard, you know? Like it needs to be like mature enough, individual disciplines of organic synthesis, of like geochemistry and physics, and then you need to put these things together to like address the big problem. I think that's the reason.
Exactly, yeah. So, I mean, you made the point that the reason you are coming up with these ideas, these results, is because you are working in a community of interdisciplinary
Exactly.
science.
That was my
And
first
this
slide.
Is really,
right?
if you are going to give one lesson to the students, you better study physics and then go to biology and then back to computer
Exactly.
science.
And then you go to philosophy in the end.
What we call in the United States convergent scientists. All of my students are convergent scientists. And they are so difficult. They work so hard, but then the rest of their life is just heaven. But anyway, here, back to my question.
Okay.
So I mentioned to you
Now I take my guard.
I mentioned to you that all of these molecules, especially proteins and peptides,
Right.
have a layer of almost
Water shell.
ice crystals.
Water shells, yeah.
Hydration shell is not a
Right.
randomly organized water molecules.
Right.
They're so incredibly specific that they probably follow the structure, physics, chemistry, and biology at the same time. So life doesn't exist under no water conditions.
Exactly.
So the water actually, in my opinion, if you can, I think you are very close to figuring that out, as far as I can see. If you could actually incorporate the effect of water here.
Right.
When there is a molecule that's left-handed,
Right.
what is the structure of the water?
Right.
Is that also left-handed or right-handed? Because that's going to produce a physical-chemical surface.
What do you mean by right-handed water?
It's a crystal structure.
Okay.
So
Is it enantiomorphous?
Molecule can grow just like your molecules there, right? So
Right.
the water molecules. Just imagine water molecules.
Right.
Okay. Here is the thing. You said what was the name of this conference?
Solvay? No?
Huh? Meeting of
Meeting of
minds.
minds, no,
Since
no.
it's meeting of minds, he is just asking me 1, 2 words, and my mind is actually meeting his mind, and we are creating at the same time.
Right.
Right, Ferhan? So here's the thing. So your chiral molecule over there
Yeah.
is made up. If you just look at the carbon
Yeah.
in the backbone, it actually has a tetrahedral structure.
Mm-hmm. Yeah, it's a sp3 carbon,
Right.
as I learned from him.
Okay, sp3. It's a perfect sp3, right? If it's in a molecule, it's not perfect, but
Right.
it's still a tetrahedral. But water molecule is also tetrahedral.
Is it really?
Of course.
Okay. Okay. How come?
So you
It
can
wasn't
have a
that like
left-handed
it
water.
been like this. Yeah.
I'm just claiming this is the first time ever.
Look,
There is a left-handed water structure on the surface of a molecule, a given molecule,
Right.
versus a right-handed water
look,
structure.
look,
Ice structure I'm talking
Yeah.
about. I'm not talking about, this is not a water in glass of in a glass.
right,
It's a water on the surface.
right,
We will discuss this in more detail.
right. I think what we would agree is we need a spectroscopist, right? And a transient
No
spectroscopist.
problem.
Yeah?
No problem at all.
Okay.
No problem. You tell us.
But it needs to have some like chiral dichroism feature, right? Like it needs to look at the transient CD spectra nearby a surface. We need to do like some femtosecond spectroscopy.
Piece of cake.
Okay.
Piece of cake.
Furkan,
Let me show you.
Yeah.
Furkan. So that CD spectra,
right?
I'll show you when after tea or something. I have exactly the same spectra.
Okay.
I've never published it. I have a right-handed peptide
Yeah.
organizing on graphene, producing one CD. I have a left-handed peptide, okay, again organizing, self-assembling on graphene, and it has the same,
Okay.
the lower.
Okay.
So what we have not looked at is how is the peptide itself when it organizes in two-dimensional crystal structures, recognizing graphene
Right.
as a left-handed chiral organization
yep.
or right-handed chiral organization.
Okay.
So that is
That's
the
interesting.
first
Yeah.
thing that I'm going to do. maybe
Okay.
tonight.
When you get back. I mean, what I argue is like, I mean, this is like a food for thought for chemists. What I argue is anytime two molecules come together, right? This is a chiral molecule. This is a chiral molecule. Whenever two molecules come together, what you see is charge reorganization, right? It's polarization. That is also in molecular recognition, right? Biorecognition involves an RNA recognizing a peptide or whatever it is. Whenever charge reorganization happens in a chiral medium, you have spin polarization because of CISS effect. I argue that biorecognition needs to consider spin exchange interactions.
I got all of that. That's beautiful.
Yeah, it's a food for thought.
No. This is just beautiful. There are two things. One,
Yeah.
what is the role of water right in the middle? Okay. What is at the end of the day, what is the overall energy after all of this? Is the
Okay.
energy being minimized? If the energy is minimized, the reaction going in one direction,
Yeah,
things are growing.
let's discuss.
Okay,
Yeah,
I'm
thank you.
sorry, I took so
No,
much time.
hocam, more questions or?
You have a question, maybe the last question, because we have also the buses department.
Okay, yeah, and people have finals and stuff.
So I have a final too, but I cannot keep myself from asking this. You did interdepartmental research, which
Right.
you graduated from physics, and your research involves chemistry, biology,
Right.
geology. So, and we are coming from similar backgrounds. I mean, we are UCAN students and stuff.
Right.
So I'm wondering how you develop yourself to like go under this research, and what's the process is like for, because I kind of feel we're on the same page and
Right.
following your footsteps, and I just wonder how is this process is like.
Perfect. I think that's a great meeting of minds question, isn't it? Okay, I mean, one thing is you need to follow your passions, right? Like I did 5.5 years of PhD, which is pretty standard for US. And I started like working on this problem only in like 3 years in. And I kind of like put behind what I have done before, which was related to quantum computing and etc., because I thought it wasn't novel enough. Like I was working on a problem in a big group, and I was like doing whatever like the group was pursuing, that was decided much before my time. That is like not fun, in my opinion. Like what I wanted to do is like I want to like pick a problem, work on it, and kind of either progress it in a degree that I'm happy with, or it's just, you know, however much progress I make. But I want to have a PhD on, which is like my idea, right? Because like if I'm not going to pursue my ideas in my PhD, like when am I going to do it, right? So that was like one thing. So you need to like pursue like what you want to do, and then after that, like both you need to like be brave enough to like look into like other fields, but also you need to be like grounded enough to work with like good experts in individual disciplines. So like I will never be a good organic chemist. Never. I'm not going to do synthesis. It's not going to happen. And John Sutherland is one of my closest collaborators. He's never going to be a physicist. Whenever we talk, I learn chemistry from him. He learns physics from me. Whenever he publishes or wants to publish a physical chemistry paper, he sends me the draft. I check the equations for him. Whenever I want to publish a chemistry paper, I send to him. He comes back with a lot of red marks and complaints because he knows chemistry much better than me. And whenever like he gives okay, I'm happy with my results. So you need to be like both grounded enough to like talk to experts in, you know, that are open-minded enough just like yourself. But also, you know, you need to be able to pursue these questions because, I mean, it relates to like his point about, you know, bringing things together. Like sometimes, like it is the right time to pursue like one question, and you need to be like able to bring things together at a level of detail, which is maybe not expert level, but you know enough to combine the knowledge landscape to solve the problem, right? That is the interesting thing. Like you don't need to be an expert to understand the detail of organic chemistry that you need for your study to be able to like kind of get your hands dirty. But you always need to be aware that, you know, you need experts' opinion afterwards. So if you kind of keep that balance in the middle, you know, not get too like creative and not too like rigorous, as they say, you know, too much rigor causes rigor mortis. You can do well.
Thank you very much.
Yeah.
Let's have a group picture now, and then T. Thank you very much.
Thank you.
Wonderful.
Oh, thank you. It was incredible. Yeah.